Okay, great. So thank you everyone for joining us. Today we are going to do a very simple activity, which is the Photosynthesis Lab using the Rebel bioprinter. My name is Maya and I'm the CEO and founder of SC3D. And we started this company not too long ago. We're primarily funded by the National Science Foundation. We got the funding to basically develop the hardware and really trying to push this into the education, not only for undergraduate students, but also for the high school students as well, because we think the early exposure for the high school students will be very important. So today we have Oliver, who is the biochemical engineer in our team. And Oliver is an expert in creating all the different types of bioassays that are being used in the different labs using the Rebel bioprinter. So I'm gonna hand it over to Oliver and he will walk you through a short presentation and then the demonstration itself in terms of how the printer runs the experiments and the assay. And then we can come back for Q&A at the end. So let me now transfer this over to Oliver.
All right, hey everyone. So I'm Oliver. As Maya was saying, I'm the biochemical engineer at SC3D, and I'll be walking you guys through the photosynthesis demo today. So first of all, to recap how a bioprinter actually works, if you're familiar with a 3D printer, it works very much the same way in that there's XYZ movement. But instead of extruding plastic filament, a bioprinter will use bioink. So typically this will consist of biomaterials such as hydrogels or other biocompatible polymers that are capable of supporting cell growth. So the 3D bioprinter can actually print a structure using the hydrogels and add cells to them, and that'll eventually grow into tissue. Anyway, so over here on the right is an example used by Organovo. So they're printing individual spheroids, which are aggregates of cells and other tissues, onto sheets of biodegradable paper. So the idea is that as you print multiple layers, they'll eventually fuse together into a solid structure. So at SC3D, we've designed our own custom 3D bioprinter for educational use. So whereas commercial bioprinters can be very expensive, we designed ours to be accessible and very low cost. Here are some of the key features. We designed it to be small and lightweight, so it's desktop sized. You can move it around pretty easily. It can also support multiple users and print multiplexed assays and other prints. The bed is actually big enough to fit 4 Petri dishes on it for multiple experiments. And finally, we have a syringe extrusion system that can print gels as well as liquids. So in the sense that it's almost like an automated liquid handler. So along with our bioprinter, we also have an integrated curriculum program that incorporates using the bioprinter. It's divided into 4 tracks that are mostly categorized based on applications. So first we have biotechnology, which focuses on printing enzymes, bacteria, and their applications in industry. The second one is green technology, which is focused on things that are environmentally friendly. So for example, biofuel production. Tissue engineering discusses 3D bioprinting for medical applications like implants, grafts, and organs. And finally, the 4th track that we're developing is food technology, which covers things like food science, chocolate printing, and so on. So as I mentioned earlier, 3D bioprinting uses hydrogels, which are networks of polymer chains that are mostly made of water. These can be synthetic or naturally derived. So some common examples are alginate or agarose, which we're actually going to be using today. So for gelation, we typically take advantage of chemical or thermal cross-linking methods. So if you're familiar with alginate, that one uses chemical cross-linking with calcium ions. Whereas for agarose, we're going to be using thermal cross-linking. So over here on the right, you can see that as agarose heats up, the individual polymer chains become separated, but as it cools down, it links it together into a solid gel structure. So the idea is that if you mix biomolecules with a hydrogel and then cross-link them, you can encapsulate them into the solid gel form. So some relevant applications are drug delivery. If you encapsulate a drug compound into a hydrogel, it can act like an extended release system where over time, the drug will gradually diffuse out of your hydrogel. And also wound dressings, which provide a high moisture environment to promote healing. For today's case, we're going to be encapsulating algae into agarose. So a few basics on algae. They're simple plants that are non-flowering. They contain chlorophyll but don't have any vasculature. So no stems, roots, or leaves. They can be microscopic in size all the way up to large kelps and seaweeds, and they're autotrophs, so they undergo photosynthesis and convert carbon dioxide, energy in the form of light, and nutrients to generate biomass. So a couple of applications, since they carry out photosynthesis, they can sequester CO2, and they can also be used to produce biomass from which we can extract useful compounds like biofuels. So in order to actually quantify photosynthesis from algae, we can use a pH indicator. So in any given solution, the atmospheric carbon dioxide is in equilibrium with the dissolved carbon dioxide. And in that solution, that dissolved CO2 will be in equilibrium with carbonic acid. And this acid can give up 2 protons to yield a more acidic solution. And this indicator will pretty much indicate that change. So yellow for more acidic and purple for more basic. So the idea is that we can colorimetrically monitor algal photosynthesis from yellow to purple as it carries out photosynthesis. The first step is to actually prepare the algae culture. So we're using a chlorella species stock today. To prepare it, we just mix it with molten agarose and fill our syringes, which we've already done. And once we've prepared the syringes, we can print them using the bioprinter. So after setting it up, you just print your algae in agarose syringe, followed by the indicator solution, and then incubate for a couple of hours and wait for the color change to occur. So I'm actually going to switch over to the video view. So I've already loaded my syringe in the extruder unit. We're going to be printing on 96 well plates that I'm just going to load onto our print bed here. And then now I'll prepare the syringe for printing, which pretty much consists of extruding until liquid comes out. And then I'm also going to set the Z0 position just to ensure that the vertical position is calibrated properly. And how that's done is I just lower the extruder unit until it's pretty much almost touching the bed. And I just want to make sure that I can kind of slip a piece of paper under it. And now I'm ready to start my print. Okay, so now my algae and agarose print is done. So I'm going to go ahead and unload that syringe. And here I have my bicarbonate indicator syringe. You can see that it's kind of yellow. So I'll go ahead and load that in to our extruder. And then again, the same thing, I'm going to manually extrude to prime the syringe until I can see liquid is coming out from the needle tip. And then set the Z0 position again. And then I can start my print again. All right, so now the print is finished. So you can see how it's kind of a yellowish color, indicating that it's mostly acidic. But I printed one earlier to show the differences. And here you can see how it kind of turned a bluish purple color. So this was the basic experiment, but there are a few modifications you can make to kind of make it a more expanded module. So you can add some different conditions like varying the amount of light. So with a full light source, you would expect the same thing to happen, that the algae array would turn purple. But if you cover the array so that it doesn't receive light, you would expect no photosynthesis to occur. So the array would stay yellow. You can also vary the amount of algae or the algae concentration in each array, as well as experiment with different species to see those effects on photosynthesis. So that was pretty much it for our algae demo today, but we also have a series of other webinars on different topics. So we have an enzymatic reaction webinar, a bacterial growth and antibiotic development webinar, as well as chocolate printing and software for creating 3D models. And then if you have questions about our printer and the BioKits, as well as the curriculum, these are just some of the main features. So we have our BioKits that include the reagents that you would need to run all these different experiments. So enzymes, enzymatic degradation, photosynthesis, which is the one I ran today, bacteria, and so on. So that pretty much wraps up the demo, but if you have questions, please feel free to ask either in the group chat or if I can unmute everyone.